Okay. All right, I think we're live. Yay.
Great.
Yay, technical difficulties. Okay. Cool. Well, I think we're up and running.
Great.
So, welcome everybody. This is another webcast from Arizona Science Center. This is Nano on the Air, and I'm Ray Cameron. So, if this is your first time tuning in, this is the third in the series that we've done this sort of talk. And all of you get to interact with our guest speaker here today via the chat window that's on the side of your screen there. So feel free at any time to type in questions. We'll be able to see them here, and we'll be able to answer those for you. So all of the folk that we have asked to be here for these talks all have ties to the world of nanotechnology and nanoscience, which is the study of the small, as we like to call it. So using atoms and molecules to do incredible things and build new materials. And if you'd like more information on that, please be sure to check out www.whatisnano.org. I'm going to put the link in the description below. And if you'd like more information, please check that out. So today, we're actually very privileged to have Dr. Rachael Sirianni with us today from Barrow Neurological, and her research is looking at using nanoparticles for new tumor treatments. So thank you for being here today, Rachael.
Thank you so much.
Cool. So would you like to start off by telling us a little bit about what you do for Barrow?
Absolutely. So my background is in bioengineering and in a field called drug delivery, which is the science of getting drugs where we want them to go in the body. And it turns out that many of the drugs that we take, whether we might take it by a pill or perhaps have a doctor inject a drug into our body, those drugs will distribute through the body, and there are parts of the body that can be difficult to access. And one of those parts is the brain. The brain is very, very good at keeping stuff out. You can imagine why. It's a very delicate balance in terms of solutes and signaling factors that maintain homeostasis in the brain. And so the brain has evolved a very tightly defined network to prevent most systemically circulating agents from accessing the brain. What that means is if we want to treat disease in the brain, when we deliver drugs by conventional routes, oftentimes those drugs are not present in high quantities in the brain. We focus on brain tumors, among other diseases in the central nervous system, but specifically one of the reasons that we focus on brain tumors is that we need to deliver chemotherapeutics to treat a brain tumor. Those drugs are often very toxic to the rest of the body. So we package the drug within a polymer nanoparticle and then engineer those nanoparticles to deliver that drug specifically to the brain to treat that tumor effectively.
Wow, that's incredible. So, I mean, what's it like to work with these small sort of particles? How do you go about that?
Yeah, it's interesting that you notice, of course, they're very small. I say you can fit 1,000 of these nanoparticles within a single strand of hair, just the width. So it's the kind of thing where you can't really see them, so we see them through microscopes, and we have specialized techniques that we can measure them and understand their properties, but ultimately we're working with things that we can't see with our naked eye.
Yeah.
On the other hand, anytime you're in the lab, you're always working with your hands, and that's something that I've enjoyed about science for a long time, which is you create something, you have a test tube, maybe it changes color, maybe it gets cloudy. There's always something visual or
really
tactile.
Exciting, yeah. Cool. All right, well, we're going to go ahead and leave the question feed open for all of you, and feel free at any time to go ahead and type in your questions. So do you mind telling me a little bit about how you got into nanotechnology?
Sure. In some ways, I have to be a little bit honest that it sometimes these things just happen. So in my undergraduate degree, I was getting a degree at Arizona State University. I got interested in research and decided I wanted to start doing research in a lab, and kind of picked a lab a little bit randomly, and that's where chance comes in sometimes, that you don't really know what you want to do, but I wanted to work in a lab. So I started working in a polymeric biomaterials lab. And this is a field where you work with engineering the properties of polymers, and you create polymers by connecting together many little parts, and by changing which parts you connect together, you create materials that have different properties.
Okay.
I really enjoyed that, and I enjoyed being able to design the properties out of a material that you want for specific applications. From there, I ended up doing graduate work in a drug delivery laboratory. So this was a lab that specializes in making nanoparticles and making biomaterials for drug delivery. And you just sort of get invested in a scientific problem, and from there it takes off.
That's really cool. So how long do you think it would be before your nanoparticle tumor treatments kind of go to market? I mean, I know these things take time, right?
Oh, it's a great question. A great question, because you'll get many different answers from many different people. So the good news is that there actually are nanoparticle therapies that are on the market that have been used for depot formulations. So things that you can inject directly into the body. And that primarily has not been in the cancer field necessarily, but the exciting part about the materials that we work with is that there is a company that's working with the same platform that they're looking at metastatic prostate cancer. So that's a kind of cancer that metastasizes through the body, and those nanoparticles are in clinical trial.
Oh, wow.
So they're very similar in design to what we work with, and the hope is that by partnering with companies in the future, we actually could bring something into the clinic. The process for that can be lengthy. First, there's the preclinical research, where you test it in vivo models and in the laboratory and obtain data that suggests it's going to be effective. And then from there, you would proceed through FDA regulation and clinical trial. And then, after probably years of clinical trial, then you could hope to have a product actually approved for the market.
Oh, wow. Okay. So kind of a long process.
Yeah.
But it's promising, though.
Yeah.
I mean, these things seem really exciting. So do they then specifically target tumors or certain types of cancer?
So it depends on how we think about targeting.
Okay.
So for a plain nanoparticle, something that just encapsulates drug, and I should describe them a little bit more. So they're spheres. If you look at an image of them, you would just see a bunch of sort of spherical-looking balls, nice and smooth and shiny. And within each of those nanoparticles is drug, and that drug is dispersed throughout the polymer. So when the nanoparticle is suspended in fluid and is within the body, the drug slowly releases. And so you get what we call a controlled release of active agents in the body.
Okay.
Those nanoparticles are about 100 nanometers in diameter. And it turns out one thing that scientists discovered early on in this field is that tumors, for non-specific reasons, can accumulate nanoparticles. And this happens because they grow vasculature that's really leaky and has holes in it. And the nanoparticles can get out of the holes and land in the tumor but not get back in. This is something we call, there's a fancy term for it, it's called EPR, or enhanced permeation retention. And that means that if you have particular kinds of tumor that grow this angiogenic vasculature, that the nanoparticles will deliver preferentially to that site.
Okay.
Beyond that, we work, and my lab works primarily on targeting strategies. And this is where we might take you call it a ligand. It's a short little molecule that you would attach to the surface of the nanoparticle. And the best analogy I can think of is Velcro. Okay. It can make them sticky. So sometimes the ligands just make them stick to the tissue that you want, and they sort of slow down as they near the tissue, and that lets more drug get to the tissue that you care about.
Oh, wow.
Sometimes you attach a ligand that actually causes that nanoparticle to go inside of a cell, or perhaps move across a cell, and that's a whole other science as well.
Wow, that's amazing. Okay, I see you brought a visual aid.
I did, yeah. This is my very simplified explanation of how we make nanoparticles.
Okay, cool.
So I actually always use the same analogy, which is it's salad dressing. It's quite literally salad dressing. So if you've ever made salad dressing, you have a little bit of oil and a little bit of vinegar, and you take a whisk and you start whisking them together, and that oil breaks up into tiny droplets, and the more that you whisk, the smaller the droplets get.
Okay.
So we have the same sort of analogy here, and the way that we make the nanoparticles is actually quite similar. So this is the oil part, and the oil would have our polymer that we dissolve into it. And in the case of a hydrophobic drug or an oil-soluble drug, we put the drug in with the polymer in the oil phase. And then we have another phase that's just water. And so the color here is representing the drug. Now, when you introduce energy into the system, I don't know if you can see it or not, you get little droplets. And if I shake it up a whole bunch, the droplets get even smaller.
Get really tiny
ones. And you can imagine that the more energy that you add to the system, the smaller and smaller they get. Now, when we make salad dressing, you make those droplets so small and maybe add a little egg yolk or mayonnaise, and it stabilizes, and the droplets never form back together.
Right.
In this example, you're going to see them merge together, but what we do in the lab is we add stabilizing agents that prevent the droplets from going back together, and there you have tiny nanoparticle with drug inside.
Oh, that's crazy. That's really cool.
Yeah, it's
what a good visual.
Yeah, it's fun in the lab. Sometimes we make nanoparticles that have really bright colors, and so we'll have, beakers with blue nanoparticles and pink nanoparticles.
Nice. So it's a colorful.
Bright at least.
It's a colorful science,
yeah.
right?
We try to be.
Awesome. Okay, well, it looks like we have a couple of questions from our team here. So this is actually from our guests on the floor today, so this is really cool. All right, so the first question we have up is what sort of concerns with cross-contamination or breathing is there with when you use such small things?
Yeah, that's an interesting question, because I think in recent years, as science has moved and medicine has moved towards nanotechnology, we've been asking really important questions about safety.
Sure.
Cells interact with things differently depending on their length scale, and nanomaterials in particular can have unexpected side effects because we're not used to studying those interactions necessarily on such a small scale. And so, for example, some nanomaterials, such as solid metals or certain other kinds of nanoparticles, can be toxic to cells. We use a particular polymer that thankfully has a very good history of what we call biocompatibility.
Okay.
And that means that within the biological system that it's doing what we want it to do and that it's not doing anything we don't want it to do. In the case of nanoparticles, the question about cross-contamination or breathing, you actually could breathe these in. I'm not sure I'd quite go up to a vial of them and snort it, but people even look at these particular kinds of nanoparticles for other disease applications, such as cystic fibrosis, which affects cells in the lungs. So the idea of nanoparticles going to the lungs is not necessarily a dangerous thing in of itself. The nanoparticles, once they're within tissue, they break down by a process called hydrolysis. And this means that the water sort of chops up the polymer and it slowly degrades away into very biocompatible byproducts of lactic acid and glycolic acid.
Okay.
In terms of the second part of the question, which is actually the first part of the question, contamination.
Yeah.
When these types of materials proceed to the clinic, there's really stringent regulations about how they're manufactured and the conditions under which you manufacture them, and so they're really pure products at that point.
Okay, wow, that's cool. And that actually is was an interesting question too that relates back to our guest speaker from last week, who was talking a little bit about safety and the society and ethics and how people perceive these things. So if you missed that talk, go back and check out Jameson Wetmore's talk from last time. All right, cool. So we have another question here. It says, are these all small enough to pass, I think that means pass, through the blood-brain barrier?
Yeah, so the blood-brain barrier, let me explain a little bit about that.
Yeah.
So most of the cells in your body, we've got sort of networks of cells throughout our body that form different tissues. And if you look peripherally at capillaries, those cells have spaces between them, and that's how stuff from the blood gets into the tissue.
Okay.
In the brain, we have a specialized network of cells, and this is known as the blood-brain barrier. These cells, instead of having spaces between them, are right up next to each other, and in fact have special proteins that glue the cells together, and this forms a network known as the blood-brain barrier.
Okay.
In addition to those are called tight junctions. So in addition to tight junctions, the cells on the blood-brain barrier are also really good at kicking stuff out. So they have what we call efflux pumps, which means sometimes when molecules get in, it then becomes a substrate for a pump that actually pushes it back into blood.
Okay.
That, again, collectively is the blood-brain barrier. So the question of how nanoparticles might traverse the blood-brain barrier is something that's actually quite unresolved in the field.
Oh, wow. Okay.
We can demonstrate that nanoparticles are internalized by cells, and in certain circumstances, it's called transcytosis. That means it doesn't just go into a cell, but it moves across a cell.
Okay.
We can see evidence for transcytosis of nanoparticles. If you're asking my opinion, I don't believe they slip between cells. We see nanoparticles in the brain. We see them beyond the blood vessels, and to the best of our knowledge, that does represent either a sort of endocytosis and exocytosis, so it goes in and goes out, or it's actively transported across.
Oh, wow.
And it's again, a big question that's up for debate, and you'll get lots of different opinions depending on who you ask.
So how do you know that it's gotten in there?
It's very tricky because
Okay.
we can't see them very easily.
Right.
So you have to track the nanoparticle in some manner. There are certain kinds of microscopy techniques that are capable of detecting nanoparticles. You can also add things to the nanoparticle in order to track what's inside of it. So sometimes we can actually put in, one good example of this would be gold. So you can actually put a tiny gold nanoparticle inside of a larger polymer nanoparticle, and when that gold is inside there, it lets you see it more easily on electron microscopy. And from there, you can look at these really amazing pictures of cells where you can see the nucleus and all of the organelles, and then you see dots.
Cool.
And you can tell that those are nanoparticles undergoing endocytosis, or maybe you see them outside of the cell on the other side, and you know that one way or another they've gotten through.
Wow.
This remains a major interest for my lab. We're using different kinds of techniques to track nanoparticle distribution. So we attach very bright fluorescent quantum dots to the nanoparticle and then look at how those fluorescent molecules distribute on a cellular level. And from there we can see that we're actually tracking the movement of the nanoparticle with that attached fluorescent molecule.
That's amazing. And they just pretty much glow then?
They glow very, very bright.
That's
and
cool.
again, you'll find, as with any scientific technique, if you ask someone else, they're going to offer up different methods.
Okay.
When I was training, in my research training before I took my faculty position, one thing that we were doing was radio labeling the nanoparticle.
Okay.
So we attach a radioactive molecule to the surface,
Sure.
and then we use medical imaging, in this case positron emission tomography, to track where the radioactivity moves. And based on where the radioactivity moves, we know that that represents, some fraction of that represents where the nanoparticle has gone.
Oh, wow. That's unbelievable. Really cool. All right, looks like we have another question. Let's see. Do the gold nanoparticles stay permanently in somebody? Are they ever ejected? Can you slowly make somebody gold?
I appreciate the last part of that question. So I'm going to defer and say I'm actually don't do any work with gold nanoparticles, so I would not be qualified to say what rate they exit the body.
Okay.
I can talk about polymer nanoparticles,
Sure.
though. So the polymers are degraded slowly, and they're degraded over the course of weeks and potentially up to a month or two, depending on the properties of the polymer and how we design that. So oftentimes most of the drug gets released before they degrade, but we do know that these polymer nanoparticles, the particular kind that we work with, are eventually cleared by the body.
Okay.
I'd say as to the question of whether you can slowly make someone gold, you probably need a lot of gold to make that. So
Because we're talking nano amounts here,
yeah,
right?
it might be a little better just to decorate yourself in gold or paint yourself in gold paint or something like that.
That's awesome. So is this similar to, like, a while back, I had heard about gold nanoparticles being used as a potential cancer therapy.
Yeah.
Where it was something like that these particles would go through the leaky blood vessels of the
Yep.
tumor, and then you could hit it with some sort of light, and it
Yes.
would kind of nuke the cell from the
Yes.
inside. Is that still a therapy that people are looking at, or is it kind of going more towards the polymer nanocapsules?
I think you find that everybody's kind of working on their own track and pushing forward in independent manner, but we also learn things from one another. The particular example that you're providing is an example of sort of triggered cell killing. So you're using an external ultrasound or UV light or other external stimulus where you can deliver your material across a tissue, but then you focus that external stimulus in order to kill selectively within the tumor and protect healthy cells. There's actually crossover in the polymer field as well. So many people are working on, we call these smart biomaterials, and they're smart because they can do what we tell them to do in essence. And so you can design biomaterials to respond to external stimuli. One very good example of this would be pH changes. So some materials have different properties in different pHs, and it turns out that the tumor tends to have a different pH than the rest of the body, and so you can use that in order to selectively release drug or cause nanoparticle retention within the tumor. Another really good example is enzymatically triggered release. And so you can design nanoparticles to have targeting shells or to have structures that are degraded selectively by enzymes that we find in that tumor microenvironment. And again, in that case, we see selective killing of the tumor relative to healthy cells.
Okay. Wow. That's really cool. So it seems like everybody's kind of working collaboratively, just kind of from different
Many
angles with
approaches,
this.
many
Yeah.
approaches. And I should clarify, when I say polymer nanoparticles, my group works with a very particular kind of polymer.
Okay.
And even within polymer nanoparticles, you could find certainly dozens of commonly used polymers, but beyond that, possibly limitless in terms of what you could imagine designing. So there are as many ways to deliver a drug as you could come up with in your mind.
That's
yeah.
really cool. So that seems kind of like it seems kind of futuristic now, but it really seems like it's becoming more of a mainstream thing as people are jumping on more research.
And in fact, I should further clarify, there are polymer modified drugs that are actually in clinical use. And so sometimes we use a polymer and we attach it to that drug to change the properties of the drug.
Wow.
As an example, many chemotherapeutics are very hydrophobic. That means they're oil loving or fat loving. And so when a drug is hydrophobic, it tends to go to particular parts in the body. And if we want to prolong how that drug circulates and increase its availability, you can attach a hydrophilic moiety to it. So people often use a polymer called polyethylene glycol to do this.
Sure.
So you link the drug to polyethylene glycol, and now it circulates for a longer period of time. There are other kinds of nanomaterials, again, not that we specialize in, but that other groups research that actually are in clinical use to treat disease in humans.
That's super cool. Wow. All right. So it looks like we have time for a couple more questions here. Let's see. Would these minimize side effects of chemotherapy, like smaller, more targeted doses, rather than the blanketed effect of chemo now?
Absolutely. I would say this is a major motivation behind what we do. If you think about what chemotherapy is doing, we're basically delivering something that's pretty darn toxic.
Yeah.
And we're delivering dose turns out to be very important. Oftentimes these drugs will show some selectivity for cancer cells because cancer cells are dividing very rapidly, and so they're more susceptible to agents that are cytotoxic, toxic to cells. But that also means, as everyone is well aware, that these chemotherapeutics tend to be also toxic to other rapidly dividing cells, such as those that grow your hair, your GI tract, etc. So you're always, in chemotherapy, you're always dosing very carefully to get at that point at which it's toxic to the cancer cells and to try to avoid toxicity to the body. So this is absolutely what we hope to achieve with targeted delivery, is that if you could increase the dose that's delivered selectively to the cancer, you decrease the total dose that you need to deliver to the entire body. You also hope that you can improve tolerability, for example. And we've shown this in our work that if you take an agent that is particularly toxic, when you encapsulate that agent, the drug is protected. And so the body doesn't see the entire dose of drug at once. And that means that you can often dose higher than you would be able to for a free drug as well.
Okay. Wow. So that's a really good thing then.
Yes,
drugs that
that's
don't
always
make
the
people
hope.
sick. Yeah.
Yes, it's always, you want it to be more potent in your target tissue and less toxic to the rest of the body.
Yeah, for sure. Okay, well, I think that's about all we have time for today, but I want to thank you again for being here.
Oh, thank you.
This was really, really fun. And so if you have questions that didn't get answered, go ahead and please keep posting them. And I will get those to Rachel, and we'll post them in the description box below. I'd like to send a really quick thank you to our friends at the NISENet, the Nanoscale Informal Science Education Network. Without their mini grant, we wouldn't be able to do these live on-air broadcasts, so thank you very much to the NISENet. And again, thank you to Rachel and all of our speakers for being with us, hanging out with us. And a huge thank you to all of you guys for hanging out, checking out our feed. Please stay tuned. We're going to be doing another one of these talks, but it is going to be postponed for just a little bit because our next speaker isn't available for the next couple of months. So stay tuned, check out our Google Plus page and our YouTube channel, and we'll try and keep you posted when that's going to be coming about. That one will be a little bit different format. We'll be asking you to give your questions ahead of time because we won't be able to do a live broadcast. We're actually going to take a field trip to her lab. So anyway, keep your eyes on the Google Plus page, and until next time, never stop wondering. All right. Cool. All right. I think we got it.
Oh.